You're sitting at your desk at home and look down at your phone. It's 10 p.m. at night and you're rushing to study for your big biology exam the next day. Fighting against your need for sleep, you're feeling tired and most of all stressed out. Stress is a universal feeling that can be expressed in the lives of children, teens, and adults. But what is stress and how is it caused? Stress is pressure or tension that one experiences in the face of something threatening or worrying. Several structures in the nervous and endocrine system work together to produce the stress response, but some key regulators are often overlooked and require more research. One of these regions is the bed nucleus of the striatum terminalis, or the BNST. The BNST is a structure in the brain that is a part of the limbic system, a series of structures that manage emotion and behavior. The BNST helps regulate stress responses, and one way it does this is by helping activate the hypothalamic pituitary adrenal axis, or the HPA axis, which is one of the most important pathways for producing stress. But before looking at the BNST's role in this pathway, there must be a stressor. A stressor could be anything in one's environment that causes stress. The sensory information of a stressor is sent to our brain to be processed by the amygdala, which confirms whether this is stressful. If it is, the amygdala sends signals to several brain regions, including the BNST. As a result, the BNST turns on specific neurons to send signals to the paraventricular nucleus of the hypothalamus, aka the PVN. From there, the PVN begins the HPA axis, which ends up producing cortisol, the stress hormone. Once cortisol is released in the bloodstream, it helps trigger physiological responses such as dilated pupils, increased breathing, and a heart rate, which are indicators of stress. This shows that the BNST helps activate the HPA axis through the PVN, but isn't fully determined which neurons deliver signals to the PVN, as the BNST maintains many types of neurons. To find which neurons project, we can use a process called immunohistochemistry, or IHC. IHC uses antibodies that are tagged with a fluorescent dye. The antibodies are added to brain regions where they'll bind to specific proteins inside of neurons to make them fluoresce. So, if BNST neurons turn on because of stress, they will glow a bright color because of IHC. Depending on which neurons glow, we could get a good idea on which neuronal types are sending signals to the PVN to activate stress. We want to identify which BNST neurons help activate the HPA axis because these neurons may be potential targets for medicine that alleviate chronic stress, which is extended periods of stress. Chronic stress has many harmful effects because it may induce psychiatric diseases or drug-seeking behaviors that lead to addiction. By accurately finding and turning off stress-regulating BNST neurons, we can attempt to lower chronic stress levels therapeutically. This is why the BNST is a significant brain region that should be studied more, so that when you do your own late-night studying, you understand the processes behind stress.